Welcome to the Funnel Effect. Today, we are undertaking a truly massive deep dive, one that reaches into the very core mechanics of what keeps the most vital and ceaseless organ in your body running, the heart's genetic powerhouse. We are dissecting a foundational scientific statement, one that fundamentally reframes how the medical community looks at cardiovascular disease, moving the conversation from broad risk factors down to microscopic genetic control.
It's a monumental reframing, and it's coming directly from the American Heart Association. Our deep dive today is based on the comprehensive Mitochondrial Genetics in Cardiovascular Health and Disease, a scientific statement from the American Heart Association. It was published in Circulation in 2025, and this document is really the result of years of synthesis. It was led by Jessica L. Fetterman and Patrick F. Chinnery, alongside a vast and critical team of contributors: Rebecca McClellan, Douglas C. Wallace, Anusul Malanan, Tina Olhala, Samantha C. Lewis, and Scott W. Ballinger. It really serves as the new gold standard for understanding this incredibly complex field.
Okay, let's unpack this with a truly shocking fact that immediately highlights the stakes and the sheer power required by this organ. Here's the hook for you. The human heart, beating continuously day and night, consumes approximately 6 grams of ATP, that's cellular energy, every single day.
6 grams of ATP, adenosine triphosphate. And to put that sheer energy demand into perspective for you, that's roughly the entire weight of a thimble full of water, just produced constantly, hour after hour. If that 6-gram energy supply were to falter for even a moment, the heart would fail.
And the crucial insight which this scientific statement anchors is that the overwhelming majority of that immense energy supply, we're talking upwards of 90%, is generated through oxidative phosphorylation, or OXPHOS. And that takes place exclusively within the mitochondria. This makes mitochondrial function and, critically, mitochondrial genetics absolutely indispensable to cardiac health.
Indeed. The source material states up front that while metabolic and genetic abnormalities have long been linked to cardiovascular disease, the specific contribution of mitochondrial DNA, or mtDNA variation, has historically been, well, complex to study. And because of that, it's been severely underrepresented in large population cohorts.
So our mission today is to crack open that complexity. We want to outline the current state of understanding, highlight where the major knowledge gaps still exist, because this is a very young field, and, most excitingly, explore how new technologies are finally changing the game. We're talking next-generation sequencing, CRISPR-adjacent gene editing, and advanced stem cell models. All these things are offering new opportunities for precise diagnosis and, hopefully, tailored treatment. To truly understand why the heart is so vulnerable to mitochondrial problems, we have to start at the fundamental biological level, genetic control. When we think of genetics, we instantly picture the nuclear genome, the nDNA, sitting safe in the cell's command center. But mitochondrial function is governed by dual genetic control.
That's the critical starting point. The entire process of cellular respiration, which produces that vital 6 grams of ATP, relies on this intricate, coordinated collaboration between the enormous nuclear genome and the tiny, separate mitochondrial genome. It's like running a multinational corporation where key operational blueprints are kept in a tiny, decentralized office.
Let's focus on that small, decentralized office first, the mtDNA. What exactly does that circular chromosome, only about 16,569 base pairs long, what does it actually encode?
It encodes the bare essentials for the OXPHOS energy generation process. Specifically, the mtDNA codes for core subunits of complexes 1A, 3 and V. But perhaps more important than the structural proteins, it also encodes the functional RNAs necessary for their expression. That's 22 transfer RNAs, or tRNAs, and 2 ribosomal RNAs, or rRNAs. These are the foundational components of the translation machinery needed to build the proteins in the complexes.
So the mtDNA holds the proprietary secrets, the blueprints for the essential moving parts of the energy factory. Now, where does the massive nuclear genome fit in?
The nuclear genome is the massive external support and management structure. It is responsible for encoding OXPHOS complex 2 in its entirety, which is a key enzyme in the Krebs cycle. And crucially, the nDNA encodes hundreds of additional subunits for the other 4 complexes, plus all the necessary assembly factors.
And beyond just the physical components of OXPHOS, the nDNA also handles the entire logistical infrastructure, correct?
Precisely. It codes for the proteins responsible for mtDNA replication, transcription, transport mechanisms that shuttle materials across the mitochondrial membranes, proteins involved in apoptosis, that's programmed cell death, and the critical quality control system known as mitophagy.
Let's pause on mitophagy for a moment. Instead of saying it's the targeted clearance of damaged mitochondria, let's make that more accessible. How does the nuclear genome manage this quality control?
Think of it as a biological recycling system. The nDNA has the ability to tag damaged, inefficient, or mutated mitochondria using the ubiquitin system and then physically clear them out. It's the ultimate quality control mechanism managed by the main office to prevent faulty energy units from poisoning the rest of the cell. The point is, you need exquisite coordination between these two genetically distinct systems: the internal blueprints from the mtDNA and the external assembly line and management from the nDNA for optimal ATP synthesis.
That coordination is complex enough, but the variation within the mtDNA itself adds yet another layer. And this brings us to the core concepts of homoplasmy and heteroplasmy.
This is a huge concept that distinguishes mitochondrial genetics from almost all nuclear genetics. Remember, mtDNA is multi-copy. There are hundreds of thousands of copies per cell, bundled into structures called nucleoids.
So when we discuss homoplasmy, what does that mean in terms of genetic stability?
Homoplasmy means every single copy of mtDNA in that cell, or even the whole organism, carries the exact same genetic variant. The variant allele frequency, or VAF, is 100%. These are typically stable, common, population-level polymorphisms that define our mitochondrial haplogroups, reflecting ancient ancestral migration.
But the real complexity and the driving force behind many mitochondrial diseases and common age-related cardiac risks lies in heteroplasmy.
Right, heteroplasmy is a state of genetic mosaicism. It means different mtDNA copies carry different variants within the same cell or even within a single mitochondria, so the VAF is less than 100%. These variants can be maternally inherited and present from birth, or they can be somatic, meaning they're acquired randomly over a lifetime due to, say, oxidative stress or replication errors.
The source material highlights a crucial link here. Heteroplasmy isn't static. Why is its dynamic nature so critical to understanding aging and heart disease?
It's dynamic because the variants accumulate, and they generally increase with advancing age. This slow accumulation of somatic mutations over the lifespan is thought to be a fundamental mechanism contributing to prevalent aging-related cardiovascular diseases like heart failure or certain arrhythmias. The longer you live, the higher the percentage of faulty energy blueprints you accumulate.
Let's discuss the non-Mendelian principles that govern how these variants express disease. We need to define the biochemical threshold.
Right, because the cell maintains hundreds, if not thousands, of mitochondria, it has a functional reserve. Pathogenic heteroplasmic variants have to reach a specific VAF threshold before the aggregate output of the mitochondria actually drops enough to impair cellular function and cause disease.
So if a patient has a pathogenic variant, say one affecting a tRNA gene at 50% VAF, they might be perfectly fine. But if, due to chance or age, it hits 65% VAF, the functional reserve runs out and the heart fails.
Precisely. For variants in tRNA encoding genes, the threshold is often cited as being above 60%, but it's highly variable. The specific threshold depends entirely on the tissue involved. The heart, being the highest energy consumer, is often the most sensitive, and the specific variant's severity. This threshold is what determines the disease onset and its severity. It acts like a catastrophic cliff edge rather than a gradual decline.
And how do VAF levels change between generations or even between cells in the same person? This brings us to the concepts of selection and segregation.
The VAF levels are determined by the dynamic interplay between selection pressures and random segregation, and they behave differently in different tissues.
Okay, so for proliferating cells, which divide frequently, like our blood cells, how does segregation affect the variants?
This is where the mtDNA bottleneck effect comes into play, one of the more fascinating biological phenomena. During the development of the egg cell line, the number of mtDNA copies temporarily reduces, and when the cell divides, heteroplasmic variants segregate randomly into daughter cells. Imagine drawing marbles from a jar. One daughter cell might get a high proportion of bad copies, another might get very few. This results in wide variations in VAFs between siblings, explaining how a maternally inherited variant can cause severe early-onset disease in one child, but be essentially asymptomatic in a cousin.
But heart muscle cells, the cardiomyocytes, are different. They're non-proliferative. They don't divide once we reach early childhood maturity. How does the VAF change in the heart?
In these vital postmitotic cells, the standard cell cycle controls for DNA replication are lifted. This phenomenon is often termed relaxed replication. Over time, damaged or dysfunctional mtDNA copies may replicate preferentially or simply fail to be cleared efficiently by mitophagy. This allows somatic mutations and heteroplasmic variants to accumulate slowly and expand, contributing directly to age-related cardiac dysfunction, independent of the bottleneck effect we see in dividing cells.
And finally, let's tie this back to deep ancestry and haplogroups.
Haplogroups are defined by collections of homoplasmic variants present at 100% VAF, which group people based on prehistoric maternal migration patterns. While they don't cause acute disease themselves, the medical relevance is that these specific haplogroups are consistently associated with overall CVD risk factors, like hypertension, and they can profoundly modify the penetrance of other pathogenic variants, both mitochondrial and nuclear. It suggests that your ancient maternal ancestry subtly influences your heart's inherent resilience.
We've established that mtDNA is complex. It's multi-copy, dynamic, heteroplasmic, and often operates right at a biochemical cliff edge. The next hurdle, and the reason this field was historically stunted, is the technical difficulty of actually finding and accurately quantifying these variants,
Yeah.
especially the low-level heteroplasmic ones using sequencing technology.
It was a massive methodological barrier.
Definitely.
The source document notes that traditional population studies were limited to analyzing maybe a handful of known variants or relying on small, clinically defined cohorts. Next-generation sequencing, NGS, and whole genome sequencing, WGS, have broken that barrier, but only if researchers follow an extremely rigorous, multi-step quality control pipeline.
Okay, let's start with the most basic difference, sequencing depth. If I sequence the nuclear genome at 30-fold depth, or 30x, that's generally considered reliable for finding a nuclear variant because I only have 2 copies of each chromosome. Why is that laughably insufficient for mtDNA?
It's a fundamental mathematical difference. The nuclear genome has 2 copies. The mitochondrial genome has potentially thousands. If you're aiming to detect a heteroplasmic variant that's present at just 5% VAF, meaning 5 out of 100 copies carry the mutation, a standard 30x depth means you'd only see that variant perhaps once or twice in your reads, if at all. That single observation is indistinguishable from a random sequencing error, which are also often in the 1 to 2% range.
So to get statistical certainty that that 5% reading is real, you need hundreds of independent reads confirming it.
Exactly. The scientific statement strongly recommends that to accurately detect heteroplasmic variants, particularly those lurking at low VAF, say 3 to 10%, you need an extraordinary sequencing depth of 500x to 1000x. This necessity completely changes the cost and computational demands of mitochondrial genomics compared to nuclear genomics.
And even with that 1000x depth, the second challenge is defining the VAF threshold itself. What percentage truly matters?
Well, there's currently no consensus on what VAF threshold should be used to define a variant as biologically real versus a technical artifact. Researchers have to set this threshold based on the sequencing platform's known error rates. If your machine has a 1% error rate, you need to set your VAF threshold well above that, maybe 3 or 5%, just to ensure you're not reporting polymerase errors as pathogenic mutations. It requires extreme caution.
And this leads us to what the source document describes as the most insidious technical problem when sequencing mtDNA contamination from nuMMTs. What exactly are these?
NoMT stands for nuclear mtDNA segments. These are ancient non-functional fragments of the mitochondrial genome that, over evolutionary history, have been inserted into the nuclear genome. Every one of us has them, and they can range up to nearly the entire length of the mtDNA.
So when I perform whole genome sequencing, my machine is not only reading the actual functional mtDNA in the mitochondria, but it's also pulling thousands of reads from these fossilized fragments stuck in the nucleus. Why is that such a profound problem for identifying new variants?
Because the NuMMTs often contain sequences that differ slightly from the functional mtDNA, and if you map your reads incorrectly, those NuMMT reads, which are just nuclear contaminants, can lead to the false positive identification of low-level heteroplasmic variants in the actual mtDNA. This has historically misled studies, suggesting variants exist when they were actually just nuclear junk.
I can see why this has stalled the field. If you can't trust the data, you can't draw conclusions. And the paper makes clear that just filtering for previously known NuMMTs isn't enough.
That's a critical methodological point. Filtering only known population-level annotated NuMMTs is insufficient because many are rare, private, or unique to an individual. The best practice solution, which requires some pretty sophisticated bioinformatics, is to identify and filter the NuMMT-derived reads before or during the initial read alignment stage.
How do you do that?
Researchers need methods to exclude read pairs that map simultaneously to both the nuclear and mitochondrial genomes, often using careful analysis of mapping quality scores. It's an intensive process, but without it, the risk of false positives at the low VAFs we care about, the ones associated with common CVD, is just too high.
Speaking of artifacts and bias, let's address the issue of reference genome bias. The whole community relied on one specific sequence, the RCRS.
Yes, the Revised Cambridge Reference Sequence, or RCRS, it served as the universal standard for numbering positions. The massive drawback, though, is that the RCRS was derived from an individual of European ancestry. If you try to map sequences from, say, African or Asian ancestries directly to the RCRS, the natural homoplasmic variants unique to those populations are treated as differences, which severely impacts read mapability and depth.
That immediately introduces a major barrier to health equity, as the document later stresses. If the reference is biased, our ability to accurately study non-European populations is compromised. So what is the recommended fix to increase sensitivity?
The recommendation is to use a sample-specific consensus reference sequence. This is computationally intensive, but very powerful. Researchers take the variants identified at a variant allele frequency of 50% or greater for that specific individual, meaning the variants that define their specific haplogroup, and use those to build a personalized reference sequence.
Then they realign the entire data set to this custom reference, essentially correcting for the person's natural ancestral variations.
Exactly. This drastically increases read mapability, boosts the effective depth, and provides greater detection sensitivity for those critical, low-level heteroplasmic variants that may be hiding in the background. While you still standardize the position numbering back to RCRS for database consistency, generating that personalized consensus reference is a massive leap forward in accuracy and equity.
This sounds incredibly complicated. Let's try to summarize the full, rigorous bioinformatic pipeline that the AHA statement now recommends for WGS data, bringing all these checks and balances together.
It's a 7-step gauntlet, really. First, you perform WGS. Second, alignment, ensuring the reference is circularized to handle the genome structure. Third, you perform stringent NumMT detection and read filtering to remove all contaminating nuclear reads. Fourth, you use variants at VAFs greater than 50% to build the sample-specific consensus reference. Fifth, you perform a second, highly sensitive round of variant calling using that personalized reference. Sixth, you standardize the physician numbering back to the RCRS. And finally, before any analysis, you apply rigorous quality-controlled procedures.
Tell us more about the specific QC filters necessary at that final stage, because even the best sequencing run has noise.
Oh, quality control is everything. Researchers must remove samples or positions with insufficient depth, again, typically less than 400 to 500-fold. You also filter out low-complexity sites, sites with poor quality base calls, and crucially, any sample flagged for contamination. Contamination is often signaled by the detection of more than one mitochondrial haplogroup in a single sample. This entire complex process is necessary to ensure that the low VAF variants you report are genuinely biological and not sequencing noise or NumMT artifacts.
And once they have that clean data, researchers need tools to interpret the biological meaning.
Yep.
Where do they go?
The statement points to several essential post-analysis tools. For population frequencies, researchers use NumAd. For known haplogroups and disease variants, MitoMap is critical. A highly specific tool is Mitotip, which provides pathogenicity scoring specifically for tRNA variants, which are notoriously difficult to assess. And finally, the field is leveraging advanced machine learning tools like Mitoscape and MCDR to help annotate and categorize these complex genetic variations. This technological infrastructure is what is allowing us to transition from just observing mitochondrial disease to actually understanding it.
The technical challenge is only worth the effort because the diseases caused by mitochondrial failure are so devastating. Mitochondrial diseases are multisystemic syndromes. They often affect the eyes, muscles, nerves, but they frequently target the highest energy consumers, the brain, and critically, the heart.
The heart is round zero. The data is striking. Approximately 30% of individuals with genetically confirmed mitochondrial disorders have associated cardiovascular issues. And here is the grim statistic that explains why cardiologists must pay attention. CVD is unequivocally the most common cause of death among adults who have these confirmed disorders.
So if a patient presents with a mitochondrial disorder, what are the most common cardiac issues a clinician should expect to find?
We see a very high prevalence of both cardiomyopathies and arrhythmias. Cardiomyopathy affects between 29 and 40% of patients. But electrical abnormalities, often detectable by a simple ECG, are even more common, affecting 39 to 68% of patients across the spectrum of these disorders.
Let's focus on the structural issues first, the cardiomyopathies. Which type dominates the presentation?
Hypertrophic cardiomyopathy, or HCM, is the most common structural presentation. It involves the thickening of the heart muscle. This can manifest very early, sometimes even prenatally, And it's important to note that this hypertrophy is often progressive, eventually leading to systolic dysfunction, left ventricular decompensation, and transitioning into dilated cardiomyopathy, or DCM, where the chamber stretches and thins. We also see rare presentations like restrictive cardiomyopathy, RCM.
And the electrical system, what kind of conduction defects are common?
The list is long, which indicates broad dysfunction across the heart's wiring. We see various degrees of atrioventricular conduction defects, severe heart block, the rapid rhythms associated with Wolff-Parkinson-White syndrome, or WPW, supraventricular tachycardia, SVT, and ventricular tachycardia, VT. All these dramatically increase the risk of sudden cardiac death, SCD.
Now let's look at some specific high-profile syndromes caused by mtDNA variants, starting with the classic.
That would be Mellis mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes. This syndrome is overwhelming, dominated by a single variant, the MTTL1 METO.3243AG variant, which accounts for about 80% of all cases.
What are the specific cardiac signs in Mellis patients?
They typically present with HCM, RCM, and hypertrabeculation, a condition where the muscle looks spongier than normal. Given the risk, the statement recommends that these patients receive regular cardiac screening, specifically every 3 to 5 years, even if asymptomatic. What's fascinating, again, is the VAF. High VAFs, 50 to 80%, cause the severe neurological Mellis syndrome, but lower VAFs, 10 to 40%, often present as the milder metabolic condition called maternally inherited diabetes and deafness, or MIDD.
Next, let's discuss syndromes caused by large catastrophic dilutions in the mtDNA, like Kearns-Sayre syndrome, KSS.
KSS is characterized by ophthalmoplegia, ataxia, and short stature, and it's typically caused by a large, single sporadic mtDNA deletion. Cardiac involvement is severe and often lethal. KSS strongly predisposes patients to severe atrioventricular conduction defects, meaning the signal from the top chambers to the bottom chambers is impaired. This defect is a major driver of syncope or fainting and, tragically, sudden cardiac death, which has been reported in up to 20% of cases. These patients often require early pacemaker placement.
And then the surprise entry, Leber hereditary optic neuropathy, or LHON, which is primarily a vision disorder, but the cardiac overlap is alarming.
It is. LHON is the most common cause of inherited blindness in young adults, usually caused by variants in complex eye genes like M.117-CHGA and MTND4. While it focuses on the eyes, the source notes that the mortality risk for LHON patients is doubled in adulthood compared to the general population. The cardiac features include HCM, hypertrabeculation, WPW syndrome, and SCD. This just proves that a single mitochondrial defect can have wildly disparate yet equally life-threatening effects on seemingly unrelated organs.
Now, let's pivot to conditions where the defect lies in the huge nuclear genome, the nDNA, but the resulting disease is profoundly mitochondrial and cardiac.
A classic example is Barth syndrome. This is an X-linked disorder caused by variants in the TEV-Zinzin gene, which is essential for synthesizing cardiolipin, a critical lipid for mitochondrial membrane structure and function. It often manifests in infancy with severe cardiac issues, dilated cardiomyopathy, hypertrabeculation, and high risk of arrhythmias, including long QT, WTW, and SCD. The prognosis is challenging. Survival is about 70%, with a mean life expectancy of around 40 years.
You also have a condition, PPA2 deficiency, that comes with a sudden presentation and one of the most urgent counseling points for clinicians.
Yes, PPA2 deficiency causes sudden cardiac failure, ventricular tachycardia, VT, and dilated cardiomyopathy, often presenting catastrophically. The crucial counseling point, which must be immediately communicated to patients and family members, is extreme alcohol hypersensitivity. The source explicitly warns that even minimal alcohol consumption can precipitate acute heart failure and sudden cardiac arrest. This is a life-saving piece of genetic information that requires immediate lifestyle modification.
Wow. And finally, a rare condition that stabilizes, DCMA syndrome.
Dilated cardiomyopathy with ataxia syndrome caused by variants in the DNA JC19 gene. This presents as early-onset DCM with hypertrabeculation. It is life-threatening very early in life, with most deaths occurring before 15 months, yet a subset of patients may stabilize by 5 or 6 years of age, demonstrating the variable progression inherent in mitochondrial disorders.
Given this enormous range in variability from severe infancy-onset DCM to adult-onset MELS, how should clinicians navigate the genetic testing landscape? Standard cardiomyopathy panels are likely inadequate.
They are inadequate because they often exclude the necessary mitochondrial genes, both the mtDNA and the relevant nuclear encoded genes. The AHA statement recommends that the first-line genetic testing for suspected mitochondrial disease should be mtDNA analysis, followed by whole exome sequencing, or WES, or whole genome sequencing, WGS. It has to be a comprehensive approach, because the phenotype variability means you simply cannot predict which specific gene will be affected.
And regarding management, since mitochondrial-specific cures are still in clinical trials, the focus remains on organized, symptom-based care.
Exactly. The clinical care requires tightly coordinated specialty expertise, cardiology, neurology, and genetics working together. Regular cardiac evaluations, ECG, ambulatory monitoring, echocardiography, and often cardiac MRI, which is becoming increasingly valuable, are essential, even when the patient is asymptomatic. While research into mitochondrial-targeted interventions is ongoing, the definitive evidence still supports conventional symptom management, HF therapies, pacing, and ICDs over unproven mitochondrial therapies for now.
We've established the role of mtDNA in rare high-penetrance syndromes. But the question that affects millions of people is far broader. How does the subtle, low-level variation we accumulate over time contribute to common, prevalent diseases like hypertension, heart attack, and stroke?
This is where the field shifts from Mendelian disorders to population genomics, and the connection is strong. The paper emphasizes that established CVD risk factors like chronic hypertension, diabetes, smoking, and just advancing age are directly linked to chronic mitochondrial stress. This stress manifests as increased mtDNA damage, altered mtDNA copy number, and general mitochondrial dysfunction within the cardiovascular tissues.
Let's use smoking as a powerful example of an environmental stressor. What exactly happens to the mtDNA in the heart when exposed to smoke?
Studies in animal models show that exposure to combustible cigarette smoke significantly increases mtDNA damage, leading to deletions and mutations, boosting oxidative stress, and impairing OCT-CHOFS complex function in the vascular tissues and the heart. In humans, data confirms this link. Individuals with conditions like type 2 diabetes or those advancing in age show higher mtDNA damage and a lower overall mtDNA copy number in their circulating blood cells. It confirms a systemic, age-related mitochondrial stress signature.
And connecting this back to the core concept, heteroplasmy, the accumulation of multiple variants in one cell, how does this age-related accumulation directly translate to CVD risk in the general population?
Generally, higher levels of heteroplasmic variants detected in blood samples correlate strongly with advancing age and overall measurable CVD risk. The most vivid example involves a common 5 kilobase mtDNA deletion, often called M.4977DEL.
That's a large deletion. How prevalent is it, and what did researchers find in diseased hearts?
This specific deletion is massively elevated. It's found 10 to 200-fold higher in explanted hearts taken from patients with ischemic heart disease compared to healthy controls. This isn't just a slight difference. This is a catastrophic level of accumulation within the failing heart muscle.
And critically, this dilution isn't just a passive marker of damage. It's also a strong predictive factor for future risk, even when accounting for other variables.
Yes. High levels of the M.4977DEL deletion were identified as independent predictors of major adverse cardiovascular events, or MAs, in patients with coronary artery disease. While the precise causal mechanism, is the deletion causing the disease or is the disease causing the deletion, is still debated, the correlation is undeniable and serves as a powerful biomarker for prognosis.
We discussed haplogroups, the homoplasmic ancestral collections of variants in Part 1. What have large population studies revealed about their influence on common chronic CVD?
Haplogroups have been associated with a surprisingly wide range of common conditions, though researchers acknowledge some results are controversial or population-specific. They've been linked to hypertension, although the reports often conflict. More consistently, haplogroups are linked to complex atherosclerotic phenotypes and myocardial infarction risk. For example, haplogroup T has been consistently associated with increased risk of coronary artery disease. They also show associations with ischemic stroke, atrial fibrillation, and even complications following a cardiac transplant, suggesting they influence how the body handles major cardiovascular stresses.
Now let's zoom in on specific point variants, moving beyond the broad ancestral haplogroups to individual mutations found within large populations. The document provides fascinating detail on variants that impact functional RNAs.
This is a key area of discovery. mtDNA tRNA variants are frequently associated with common CVDs like CAD and hypertension. The mechanism is hypothesized to be impairment of tRNA structure and function, which destabilizes the entire process of mitochondrial protein synthesis and, consequently, OXPHOS activity. When you break the construction crew, the tRNAs, the factory OXPHOS, It grinds to a halt.
Give us some of the most compelling examples of these specific variant associations, linking a single nucleotide change to a chronic cardiac risk factor.
One of the most studied heteroplasmic variants is M.3256CT, located in the MTTL1 gene. Researchers found that its variant allele frequency correlates positively and strongly with the degree of atherosclerotic stenosis and carotid intima media thickness, even after rigorous adjustment for age and sex.
That's a clear dose-response link. The more copies of this specific variant you carry, the thicker your arterial plaques tend to be.
Exactly. Other control region variants show similar links to common risk factors. The variant M.16183AC is associated with higher systolic blood pressure and mean arterial pressure. Another variant, M.16189PC, is associated with coronary artery disease and type 2 diabetes, and it's found significantly more frequently in patients presenting with dilated cardiomyopathy. It's a nexus point, also linked to metabolic syndrome, higher triglycerides, and elevated HbA1c. What's truly illustrative of the complexity is that the association isn't always detrimental. Some mtDNA variants appear to be protective or modulating. That is the ultimate nuance of the system. While M.12315GA shows a positive correlation with increased carotid intima media thickness, suggesting risk, the variant M.13513GA actually shows a negative correlation with intima media thickness. This implies that certain maternal mitochondrial backgrounds can genuinely modulate or reduce disease susceptibility to common nuclear-driven risks. It underscores that we are looking at a complex genetic balancing act, not just a simple list of bad mutations.
The deep mechanistic understanding we now have, the connection between specific variants and plaque thickness or blood pressure, is only possible because researchers have developed increasingly sophisticated models. For a long time, the inability to edit the mtDNA genome was the great white whale of mitochondrial genetics. That barrier is falling. Let's start with animal models.
Mouse models have been essential, but limited. Researchers often had to rely on xenomitochondrial or complastic models, which involve leveraging existing mtDNA variants in different mouse strains. The core problem was, if a heart phenotype emerged, you couldn't confidently pinpoint which of the many differing mtDNA variants was the causal one.
This emphasizes the importance of understanding mitonuclear interactions, how the nuclear genome modifies the effect of the mitochondrial variants. The source provides compelling examples of this critical interplay.
These examples are crucial for demonstrating penetrance. For instance, researchers found that knocking out the nuclear gene ANT1, which codes for the mitochondrial ATP-ADP exchanger, only resulted in accelerated cardiomyopathy development when paired with a specific mtDNA background carrying the MTND6 P.25PL variant. When that same nuclear knockout was paired with a different mtDNA variant, MTCO1421VA, there was no effect on the cardiac structure. The disease is entirely dependent on the specific, precise alignment of both genomes.
And another key mouse study focused on the functional effect of the heteroplasmy threshold within the heart itself.
Yes, this model was built with a cardiac-specific mtDNA deletion. As the mice aged, their individual cardiomyocytes developed mosaicism-varying VAF levels of the deletion. This study showed that the mosaicism was sufficient to cause cardiac arrhythmias, specifically during exercise. This is a foundational mechanistic finding. It demonstrates that even if only a fraction of the heart muscle cells carry pathogenic mtDNA deletions, the collective biochemical defect is severe enough to cause clinically relevant CVD.
Beyond mammals, the pace of research benefits from simpler organisms.
Invertebrate models like the nematode worm C. elegans and the fruit fly Drosophila are highly advantageous due to their tractable genetics, ease of editing, and rapid life cycles. While C. elegans lacks a true cardiovascular system, Drosophila does possess a heart tube, making it slightly more relevant for rapid screening and genetic studies focused on specific mtDNA variants.
Moving from whole organisms to cells, the classic model for studying mtDNA has been the cybrid.
Cybrids are built to study a single variable. They're created by fusing mtDNA-depleted cancer cells, called Rho cells, with the cytoplasm or platelets from a patient. This allows researchers to study the effect of a single, defined, patient-specific mtDNA variant in a standardized, controlled nuclear background.
And cybrids have provided some of the deepest insights into that critical biochemical threshold we discussed earlier. Using cybrids with the common MLAS M.3243AG variant, what did they discover about the transition from normal function to dysfunction?
They found that VAF level affects cellular phenotype in a nuanced way. They saw that maximal respiratory function remained largely intact until the VAF crossed the steep 60% threshold. But here's the nuance. Even VAFs of 30 to 40% caused observable changes. specifically smaller cell size and changes in the mTOR signaling and growth pathways, even though the cells were still meeting their maximal respiration goals. Higher VAFs, closer to 50 or 90%, caused a strong upregulation of glycolytic enzymes, which is the cell shifting away from ALK-XPHO as it fails. This shows the pathogenic effect involves metabolic and epigenetic shifts long before the heart muscle collapses from lack of energy.
The newest and perhaps most promising cell models come from stem cell biology, induced pluripotent stem cells, or iPSCs.
Patient-derived iPSCs are generated from somatic cells, like fibroblasts. What makes them uniquely useful for mitochondrial research is that the reprogramming process itself mimics the natural mTNA bottleneck we discussed earlier.
So the technical challenge of reprogramming somatic cells actually provides a scientific benefit by creating a range of VAFs.
Precisely. The process naturally results in subclones with varying yet stable heteroplasmy levels. Researchers can then isolate and select subclones with 10%, 30%, or 60% VAFs to study the differential effects of that specific pathogenic variant on the developing heart cell.
And can these iPSCs be successfully differentiated into heart cells?
Yes, with some caveats regarding maturity. While some extremely high-level pathogenic variants can interfere with the final step of differentiating the iPSCs into actual cardiomyocytes, most can be successfully differentiated, providing a patient-specific cellular model that is highly relevant for cardiac study. They offer a unique window into how a specific variant affects patient-derived heart cells.
Finally, let's talk about the absolute frontier, mtDNA gene editing. If we can find the variant, can we fix it?
For decades, no. But the first breakthrough tools were mitochondrially targeted nucleases, such as zinc finger nucleases or tail nucleases. These are engineered proteins that are specifically transported into the mitochondria. Their function is to induce double-stranded breaks in the DNA, which leads to the selective degradation of the pathogenic mtDNA copies.
But those nucleases only work if the cell is already heteroplasmic, and they only degrade. They can't introduce new beneficial variants.
That limitation led to the second, more advanced generation, base editors. These use tail or zinc finger proteins to target deaminases to the mitochondria. This allows for A to G or C to T base editing, enabling researchers to introduce specific point sequence variations. This is a huge step forward for both therapeutic and basic mechanistic studies.
What are the current limitations of these advanced editing tools, and what is the current holy grail the field is chasing?
Well, the base editors still suffer from the introduction of bystander variants within the editing window, unintended edits happening near the target site. And the ultimate limitation, critically noted by the AHA statement, is structural. The absence of appropriate mitochondrial transporters to shuttle the necessary guide RNAs across the double mitochondrial membrane still prevents the full implementation of the game-changing CRISPR-Cas technologies for mtDNA editing. We're close, but that final hurdle of delivery remains.
Let's consolidate everything we've discussed by looking at the core mechanisms. How exactly do mtDNA variations lead to cardiomyopathy? It's not just a lack of ATP, it's a systemic breakdown.
What's fascinating here is the sheer intricacy of mitonuclear communication. Because the ALK-XPHOS machinery requires coordination between both genomes, consistent signatures of dilated and hypertrophic cardiac disease include the downregulation of mitochondrial biogenesis and defects in mitochondrial tRNAs and tRNA synthesis. When the ability to read the mtDNA blueprints, the tRNA function, is impaired, the heart muscle cannot maintain its energy machinery.
So when that communication breaks down, the heart begins to fail. Beyond gene expression, mitochondrial function is fundamentally tied to cellular metabolism and ion handling.
Right. Altered mitochondrial respiration has cascading effects far beyond energy output. It affects the supply of crucial metabolites needed for lipids, amino acids, and purines. This means the mitochondrion plays a profound epigenetic role by regulating key cofactors like acetyl-CoA and NAD+WAY, which are essential for modulating nuclear gene expression. A defect in the energy factory sends regulatory signals that change how the entire cell behaves.
And the other critical cellular function is calcium handling, the trigger for every heartbeat.
Any alteration to mitochondrial membrane potential, which is immediately affected by pathogenic mtDNA variants or lack of ALK-XPHOS, will disrupt calcium dynamics. Mitochondrial membrane potential regulates calcium uptake and storage via the mitochondrial calcium uniporter and the sodium-calcium exchanger. Dysregulation of these transporters causes poor calcium control, which is strongly associated with the pathology of heart failure and hypertrophy. Essentially, poor mitochondrial function leads to cellular excitability and eventual collapse.
Finally, let's discuss the modern mechanism that connects mitochondria not just to heart function, but to generalized vascular inflammation. The concept of mitochondria acting as damage-associated molecular patterns, or DAMPs.
This is a major area of research. Mitochondria originated as ancient bacteria, and their components retain primitive bacterial features. When the mitochondrion is damaged or stressed, say due to accumulated heteroplasmy or environmental stress like smoking, it releases components, particularly mtDNA, into the cytosol. These components act as damps, triggering an intense, sterile, inflammatory response because the cell recognizes them as foreign invaders.
Which specific inflammatory pathways are activated by this released mtDNA?
The mtDNA activates receptors like TLR9, toll-like receptor 9, and the CGA sting pathway. Activation of TLR9 initiates NF-kappa B signaling, leading to the transcription of pro-inflammatory cytokines. And this has been implicated in chronic vascular dysfunction, preeclampsia, and hypertension. The CGA sting pathway also promotes a chronic inflammatory environment strongly implicated in atherosclerosis and general CVD, often triggered by damage from factors like cigarette smoke exposure.
So, mitochondrial damage doesn't just cause heart failure from lack of energy, it triggers a chronic inflammatory state that attacks the entire vascular system, essentially turning the powerhouse into a biological danger signal.
That is the broader systemic consequence of mitochondrial dysfunction.
Despite all these mechanistic insights and new technologies, the field is still dealing with significant knowledge gaps. What are the main obstacles the AHA statement highlighted for future research?
There are four key areas demanding attention. First and foremost is the issue of ancestral diversity. Most population studies still rely on data from individuals of European ancestry. This lack of diversity severely limits our ability to identify population-specific variants and impedes health equity. We need massive efforts to increase sequencing diversity.
And what about studying the mitochondria in their native living environment rather than just in a petri dish?
Second, we have a critical lack of understanding regarding mitochondrial homeostasis, specifically how mtDNA integrity and copy number are maintained within the native cardiovascular tissues. Much of the current data relies on blood samples, which may not accurately reflect what is happening deep inside the heart muscle.
Third, what about signaling between organs?
We lack data on how myokine signaling molecules like GDF15 and FGF21, often released from organs like skeletal muscle when they undergo mitochondrial stress, affect distant organs like the heart. The communication network between the skeletal muscle, the liver, and the heart via these molecules is still poorly understood.
And the final gap relates to that core concept of variation within a person versus variation across populations.
We need much better data on how mtDNA integrity or copy number varies within the tissues of a single individual compared to between individuals. Understand this inter-individual variation is absolutely crucial for developing accurate, predictive biomarkers that can clarify why two people with the same pathogenic variant or the same environmental exposure might have dramatically different clinical outcomes.
So what does this path forward look like? Leveraging all the new technology we've discussed, the 1000x sequencing depth, the iPSCs, the base editors?
The future is bright. The new technological opportunities allow for deeply mechanistic studies that weren't possible 5 years ago, leading to vastly improved diagnostics. The ability to use massive-scale omics datasets like the Transomics for Precision Medicine or TopMed or the UK Biobank combined with machine learning is critical. This approach allows researchers to finally move past broad classifications, better classify patients with mitochondrial presentations, and ultimately tailor therapeutic approaches that target their specific genetic and metabolic profiles.
What a remarkable and comprehensive deep dive into genetic mechanisms. When you synthesize all this dense information, the main concept is absolutely clear. Mitochondrial DNA is not just a secondary player in cardiac health. It is a fundamental primary regulator of cardiovascular well-being, where tiny variants and low levels of heteroplasmy can have profound, life-altering, and long-term consequences, especially as we age.
The AHA scientific statement marks a crucial new era. We've moved past simply observing the link between energy failure and heart disease. Now we have the technological tools, the rigorous bioinformatic practices, and the sophisticated cellular models necessary to tackle these deep genetic complexities directly and begin to design targeted interventions.
Here's the final provocative thought for you to carry forward. Reflect on the profound biological implication that a person's maternally inherited mitochondrial genetics, their ancient ancestral haplogroup, and their lifetime accumulation of specific heteroplasmic variants might silently predetermine or modulate the penetrance of all their nuclear-encoded heart disease risk factors. This single small circular chromosome is quietly orchestrating the fate of your heart, often years or decades before the symptoms even appear. Thank you for watching The Funnel Effect.